Hello everyone, welcome back to the MEAB Sciences channel of Dr. Dipankar Ghosh. Today I'm going to deliver a very interesting talk on biocompatible nanoparticles and nanosheets as a potential delivery system for therapeutics, the way forward. So this particular lecture talk has been delivered as an invited speaker in National Science Day, which has been organized by Atal Bihari Vajpayee Vishwavidyalaya, Bilaspur on 20th February 2022. And the basic object or the preamble of this National Science Day celebration is on the integrated approach in science and technology for sustainable nature. I, Dr. Dipankar Ghosh, Assistant Professor, Microbial Engineering and Algal Biotechnology Laboratory, Department of Biosciences, JIS University, will deliver this talk. So let's get started. A significant effort is really underway to develop the biocompatible nanoparticles as a potential alternative for conventional nanoparticles as a therapeutic delivery system. As you know that conventional nanoparticles having a lot of demerits or pitfalls that actually attracting the biocompatible nanoparticles in the way forward. Among the various options, biocompatible nanoparticles are attractive future delivery system, especially for nucleic acid, protein, drug. The reason is that due to their smaller size in nanometer scale, lower molecular weight, higher solubility, higher permeability, higher association efficiency with the drug, and better biodegradability after the use has been done, high release efficiency of the drug which has been conjugated or entrapped within it, and do have a non-hemolytic nature, lower cytotoxicity at the normal physiological condition within the human body. However, there are significant remaining issues. They are restricting or constraining the practical application, such as specific interactions of the biocompatible nanoparticles with the human organs, tissue cells, and resistant microorganisms, including multi-drug resistance microbial pathogenic communities, especially the biomolecules and also the fate of this drug as a metabolic outlet. Hence, the further deep research for upcoming or overcoming these practical barriers may be pioneered a new avenue in the near future. That's the reason why we have been going to understand the different case studies, like how the biocompatible nanoparticles or the nanosheets could be the better way out or outlet compared to the conventional nanoparticle and make it attractive future delivery system for the drug. So based on that scenario, the current talk will be focused on brief outline and sketch of biocompatible nanoparticles and their therapeutic application, considering some case studies that have been done since few years ago. So this is the basic focus in a diagrammatic presentation as a graphical abstract, I would say. So we would like to focus on two different arenas. One is the low molecular weight biocompatible nanoparticle or the nanosheets, especially based on alginic acid and chitosan conjugates. Second scenario would be the graphene oxide para-aminobenzoic acid nanosheets. And ultimately target would be to treat the drug resistance or the multi-drug resistance bacteria like E. coli XL1, which is tetracycline resistance, as a model microorganism to carry out in vitro and in vivo characterization and validation. So this is a very simplistic workflow, the way we have carried out the experimental approaches. So I would like to explain in very brief due to the time constraint. First, in very general, whatever the nanoparticle or nanosheets are, you have to follow out certain very basic thumb cross rule. One, you have to get started with the preparation or the design of the native high molecular weight nanoparticle or nanosheet that can get dissolved within the suitable solvent, buffer, or any other forms. Then you have to carry out the in vitro depolymerization measurement, because you have to reduce the size, you have to depolymerize them. Especially here we have done the case study on alginic acid, chitosan, and graphene oxide nanosheets. So depolymerization will allow you to reduce the molecular weight. Now, if the depolymerization has been occurred perfectly, that you have to characterize, you have to check the depolymerization by the DNase assay, and you have to calculate and also measure or determine the average molecular weight of those depolymerized nanoparticles or the nanosheets, followed by characterization. So when you have done the characterization by different biophysical tools like XRD, FTIR, and XRD, or by the GPC, gel permeation chromatography, you have to carry out the next step, that how you are going to load, how you are going to conjugate with your drug of interest. Like here, our drug of interest is tetracycline, which needs to be get loaded in the low molecular weight nanoparticle or the nanosheets. Afterwards, you have to also carry out the checking or determination of physical loading efficiency of those drugs in so-called low molecular weight nanoparticle or nanosheet. In vitro depolymerization by the DNase assay, because you are using biocompatible molecules like chitosan or alginic acid. So these molecules are going to be get released by different monomeric hexose or pentose sugars that you can easily quantitate by checking the availability of the reducing sugar by the dinitrosalicylic acid, that is the DNase assay. So we see that after the hydrolysis by different treatment, chemical treatment, or by checking the effect of different physicochemical parameters, you can easily able to identify the depolymerization has been established or not by the dinitrosalicylic acid. Afterwards, when the depolymerization has been completed, then you have to check the lower molecular weight nanoparticles or the nanosheets that you have developed by following the FTIR studies or by H1 NMR or by following X-ray diffraction pattern. Then you have to carry out the determination of the molecular weight by the gel permeation chromatography that will allow you to understand how you can able to reduce the average molecular weight in the unit of kilodalton. You can here see that while we have been considering the native high molecular weight alginic acid, when it has been depolymerized, the size has been reduced to 20.95 kilodalton. Similar way, after when the nanoparticle or the nanosheets has been developed, then you have to check their size, even their surface charge, because surface charge plays a very, very important role that will allow you to understand which kind of drug you can able to conjugate or it will be get adsorbed within this nanoparticle or the nanosheet for the delivery system and their sustainability. So to do that, you have to carry out the high resolution TAME or the same and also calculate the zeta potential distribution to calculate the surface charge. Then you have to carry out the profile for the in vitro release of the drug. And it will allow you to understand how extend and how efficiently your drug will be get released when it will be applied in the in vivo conditions to treat the drug resistance microorganism by measuring the drug release percentage efficiency over time. Afterwards, as I said, you have to carry out the in vivo antibacterial activity to make sure the released drug is really working or the conjugate nanoparticle with the drug can be really effective or not. That's why you have to perform different sets of control. Like here we have take one control for the inoculated control where only E. coli XL1 tetracycline resistance microorganism do present. In the second control, we have tried out the NP without the drug conjugation. And next one is the negative control, which will allow you to understand that the medium is free of any other forms of microbial contamination. And this is the way we have able to come up with the minimal inhibitory concentration, that is 700 micrograms per ml. So this concentration is actually, we put forward when based on the nanoparticle or the nanosheet while it is being conjugated with the drug in a powdered form. Then we have also checked how this antibacterial activity has been happening within the microorganism. To perform it, to identify it, we have done the scanning electron microscopy, and we have observed that in case of the alginic acid or chitosan based conjugated tetracycline, they do actually making the pore formation or the scar formation on the polar ends of this microorganism. So if you consider this is the control, the cells are looking perfectly okay, but while we have done the treatment of this drug conjugated alginic acid or chitosan nanoparticle, it shows that some polar scar has been observed. Now we will move to the next segment of our study, that is the nanosheet application while it has been conjugated with the drug, in our case tetracycline. So it can easily seen from here. You have to carry out similar way the physicochemical characterization following FTIR and fluorescence spectra that will allow you to understand that drug is really getting conjugated or they are getting absorbed or entrapped within the nanosheets or not, followed by the characterization of your in vitro release of the drug. Also, you have to check the biocompatibility and also the image analysis, like how the nanosheets are looking like after conjugation with the tetracycline drug. So the biocompatibility assay plays very important role over here, because that will allow you to understand that the nanosheet that you have developed, whether they really get solubilized and not getting deposited within the in vivo bloodstream. So that's why we have performed this very unique assay. We have chosen the nutrient broth medium. We have also chosen the blood serum. We have also considered water and the phosphate buffer saline to make sure using four different forms of solvent. Now we have found out when we have developed this graphene oxide nanosheet, they are easily get deposited. That's why we also conjugate, or we have also reacted with the para-amino benzoic acid with the nanosheet. So while we have been treated our graphene oxide nanosheet with para-amino benzoic acid, and then we have conjugated with the tetracycline drug, then we have found out after several long period of time in terms of days, there is no deposition has been taken place. So they are easily get solubilized and not getting deposited. So it actually allow you to understand that the molecule, the drug which has been conjugated with the GO in conjunction with the PAPA, they are actually solubilizing acid as well as especially drug in our case. And that's why it would be a future way forward. So in nutshell, what I would say, the low molecular weight nanoparticles or the nanosheet have enormous potential as a drug delivery system against the drug resistance or the multi-drug resistance bacteria. In our case, tetracycline resistance E. coli XL1 strain. So these are the few references that I have worked out long time ago and have been published in few very prestigious journals. In Springer Nature, he is there as well. And finally, I'd like to acknowledge Dr. Latika Bhatia. She has invited me as a convener of this particular seminar. And I would also like to thank the organizing committee of Atal Bihari Vajpayee Vishwavidyalaya Bilaspur, Department of Biosciences, JIS University, for allowing me to carry out this deliberation in front of you and all eminent participants on this online platform. Hope you enjoy this session. And this is my small group. I've been working here since last couple of years, and we have developed a very good coordination and working culture environment in terms of research and innovation and technology transfer. So this particular lecture session, I've already made a pre-recorded session that will be get uploaded in the MEAB Sciences channel, YouTube channel that we have been operating on behalf of our MEAB Sciences Laboratory. That is Microbial Engineering and Algal Biotechnology Laboratory. Thank you very much. Hope you enjoyed this session. Take care all. Bye-bye.